Every year, more than 100,000 new patients are diagnosed with end-stage renal disease and could benefit from renal replacement therapies such as dialysis and kidney transplantation. Though offering extended survival times at lower cost and a higher quality of life, fewer than 25,000 kidney transplants are performed annually because of the significant shortage of donor organs. Because of the disparity between organ supply and demand, many patients are placed on a transplant wait list. While on the wait list, a person may wait years before an acceptable organ becomes available while their limited kidney function continues to negatively impact their life. Others, who are deemed less than ideal transplant candidates, are not waitlisted because they have virtually no chance to receive a donor kidney. As an example, more than 120,000 patients were diagnosed with end-stage renal disease in 2016, with approximately 36,000 patients waitlisted. About 36% had received a transplant by 2019. About 36% remained on the wait list in 2019. 7% had died waiting. And approximately 20% had been removed from the wait list, typically because their health had deteriorated to the point where they no longer were a viable transplant candidate. Unless transplanted, patients remain long-term on dialysis with poorer survival and negative impact on life. Chronic dialysis is not only a drain on patient lives, but also on the economy. Hemodialysis care costs the Medicare system an average of $90,000 per patient annually in the United States, for a total of $28 billion. Every year, the cost of caring for dialysis patients is about 3 times more per patient than for kidney transplant recipients. Though increasing the supply of donor organs is the only mechanism to provide every patient in need a transplantable organ, it is unclear how to extend the amount of available human organ donors to meet this need. Xenotransplantation, the transplantation of animal organs into human beings, provides an opportunity to solve this problem. Our goal at Makana Therapeutics is to make xenotransplantation a viable medical therapy. Pigs are excellent donor candidates for human beings for several reasons. First, they share many anatomical and physiological similarities with people. Second, their reproductive capacity is sufficient to allow enough organs to be produced to meet the demand. And third, they're capable of being genetically engineered so that their organs are ideally compatible with the biology of human beings. We are working to overcome the handful of barriers that have been identified as possible challenges to the clinical application of xenotransplantation. First, disease transmission between species must be prevented to ensure safety of the therapy. This concern can be mitigated by producing donor animals in a pathogen-free environment as a barrier to the introduction of infectious disease. In addition, diagnostic testing can be performed to verify that the donor animal is free of disease. Another barrier to xenotransplantation is the rapid decline in organ function post-transplant. This problem arises because patients have antibodies that vigorously attack the cells and tissues from other species, which leads to organ failure within minutes to hours. Decades ago, scientists showed that it was possible to reduce pig targets of the recipient antibodies by inactivating a single porcine gene, which slightly prolonged organ function. However, because of the challenges associated with gene editing technology and a lack of obvious additional target genes, this approach lost favor. Consequently, additional reasons to explain organ failure were suggested. These included various physiologic incompatibilities across species that were proposed to cause dysregulation of blood coagulation and inflammatory mechanisms, among others. The field focused on mitigating physiologic incompatibilities by expressing human transgenes in pigs, as this genetic engineering was far more accessible as a genetic engineering technology. When considering the role of physiologic incompatibilities as a barrier to xenotransplantation, my brother, Joe Tector, on the right, a transplant surgeon, physician scientist, and founder of Makana Therapeutics, noted that the aberrant regulation of these physiologic pathways in xenotransplants resembled pathologies observed by others in human-to-human kidney allotransplantation, where no physiologic incompatibilities exist. Because it had been previously shown that recipient antibody damage of donor tissue in allotransplantation could lead to physiologic dysregulation, he proposed that antibody-mediated damage was central to the immunologic and physiologic incompatibilities of xenotransplantation. With this insight, he made the key focus of our efforts avoiding antibody recognition of pig tissues, which makes us unique in the field of xenotransplantation. He hypothesized that by eliminating human antibody binding to pig cells, the transplanted pig kidney would not be damaged by antibody-driven mechanisms, and the dysregulation of various physiologic pathways as a downstream consequence would not occur, making the introduction of transgenes unnecessary. We expect this will simplify the regulatory path because we will not need to validate durable expression of the transgenes in the right place at the right time. In addition, chronic transgene expression may have unintended consequences. Several of the currently used transgenes are often upregulated in cancers to provide tumors with a growth advantage. Furthermore, some transgenes that have been studied in xenotransplantation have been identified as receptors for human viruses, which may increase zoonotic disease risk if expressed in a pig tissue. In the pig to non-human primate xenotransplant model, which closely approximates the pig-to-human xenotransplants, the principles of using gene-edited donors to minimize recipient antibody attack have allowed us to repeatedly achieve greater than 1 year survival with the pig kidney providing life-sustaining function. 2 of our transplants have survived more than 500 days. In the last decade, armed with the latest gene editing technologies, we have made a triple knockout pig in which we have inactivated 3 separate genes that produce molecules that are targeted by human antibodies on the surface of the pig kidney. We have further shown that the triple knockout pig can reduce or in some case eliminate all human antibody binding to tissues from these animals. We believe the triple knockout pig will be a suitable kidney donor for as many as 70% of kidney failure patients. For those currently awaiting organ transplants, 30% show no detectable antibody binding to cells collected from the triple knockout pigs. An additional 40% of patients have some antibody binding to these cells. However, those antibodies can be reduced through clinically available therapies. Those are the people who need us to figure this out. Of course, many more than that are not even counted, as the organ shortage prevents them from being put on the wait list. Because our goal is to enable every patient to benefit from a transplant, including those who are not currently waitlisted, our research has 2 goals. Bring the triple knockout to market as efficiently and safely as possible, and continue the research and development process to identify additional improvements and supportive medical therapies that will enable every patient access to transplantation. Our founder has had people being worked up in the kidney transplant program say, "Hey, are you the pig guy? Please hurry up." These anecdotes are supported by further studies suggesting general acceptance of the use of pig organs for human transplantation. In addition to improving people's quality of life, transplantation has also been shown to reduce the per patient annual cost of end-stage renal disease by almost 2/3. We estimate xenotransplantation would be roughly equivalent to, and certainly not exceed, the cost of current human-to-human transplants. Currently, the US occupies about 25% of the market related to end-stage renal disease. The global organ transplant market is predicted to exceed $60 billion by 2027. As the market grows in the future, we are fortunate that the reproductive capacity of swine and our access to strong animal husbandry will allow us to scale our product to meet demand. Our team includes several practicing transplant surgeons who have cared for thousands of transplant patients as their direct physician. We also have a team of scientists with decades of cumulative experience in genetic engineering and in producing genetically modified pigs. To facilitate our transition to clinical application, our team also includes experts in transplant diagnostics, experts in veterinary care, and people skilled in hospital administration. We believe there are no remaining scientific barriers. Therefore, as a preclinical stage company, we are moving toward our first-in-man trial as early as 2022 and could be to market as early as 2025. In order to help us successfully perform clinical trials and bring this product to market as quickly and safely as possible, we are actively pursuing additional funding, expert and regulatory issues, and other key partnerships in healthcare. In summary, We believe xenotransplantation will allow end-stage renal disease patients to receive a kidney immediately without the years of waiting and uncertainty that they currently face.